personal battle, but I believe. Um, Thank you for the reminder as always. Right. So Dr. Price received her PhD from Cornell University in 1959 and went on to develop a vaccine against Pasteurella anapestiferous virus, which is found in ducks and other waterfowl. So animals need vaccines too, class. Animals need vaccines too. So she helped fight the good fight with duck vaccination. And she also did additional research in the duck research laboratory for over 18 years and developed a number of other vaccines that are used commercially throughout the 70s. And she also worked as a microbiologist at the National Wildlife Health Center on the National Biological Service in Madison, Wisconsin, where she continued this work to fight avian diseases and develop treatments. So there are a diversity of careers out there for microbiologists. You don't just have to work in a lab, although I think that's pretty cool. You can also integrate it into a wildlife context and develop vaccines or study diseases that infect animals. So lots of opportunities there. And as always, I am happy to discuss those things during the student success hours. Speaking of which, a slight modification for tomorrow. They're going to be from 8 to 9 a.m. tomorrow only. And I'm just want to reiterate that I'm always happy to meet by appointment every week. I reserve time outside of office hours to meet with you. So if you do not find the office hour times convenient, I completely understand. We all have busy schedules, things to do, people to see, maybe not so many places to go, but hey, you can always go to student success hours. I will be there and also happy to make appointments at other times. So feel free to reach out to me on that. And if you're sitting here thinking, man, like I really want to go to office hours, but I just don't, I feel nervous. I don't know what to ask. You can literally ask me about any of the African American scientists I featured. You're like, hey, I want to learn more about them. Maybe you're like, hey, I want to learn about a Latinx scientist. Can we do some research there? Or you were mentioning careers, like how did you end up as a professor at San Jose State? Tell me your life story, because I zoned out the first time you told it for like 30 minutes during the first day of class. All I remember is you got frostbite. So you know what? That's okay. Like plenty of people zone out when I talk. I'm used to it. My friends do it. Sometimes my family does it. Like it's real. So happy to just have basic level conversations about life or class content, right? You don't have to like, but I just don't want anyone to feel like, hey, I get this material. It's pretty straightforward. You don't have to like pretend you don't get it and be like, oh, that's the difference, like between gram negative and gram positive. You don't have to do that. All right. So that's my pitch. Let's move on to the content. Awesome. Here we go. Not everyone gets to ride into the weekend with some microbiology. So it's an honor to tell you about it. Growth cycle. So we didn't finish the slides from last class, and one quick just structural thing. I always will have more slides than we get to in the slides that I post. And this is intentional because I never want to run out of material for you. I would feel horrible if we ended class 15 minutes early and then everyone was like, oh, like cool, you don't have stuff to teach us. Like, no, you're paying for this education. You deserve every minute. For those of you who enjoy getting out early, I'm sorry, like that's not my thing. All right, but that being said, we will be covering those slides in a slightly different order this class, so don't freak out. Everything should be in order in your slides that I posted, but just want to let you know. So with regards to the growth cycle, most bacteria divide by a process called binary fission, where one cell becomes two, right? It's like, it's probably one of the oldest magic tricks that exists, except it's not a magic trick, it's science. And so these cells will replicate through this division, and we've touched on this at various points in the previous lectures. However, it is important to note that some cells or microbes are able to divide asymmetrically. And we saw some examples of this with Caulobacter crescentium, where you have a stalk cell and a flagellated cell. And you can also have budding cells where you don't necessarily have flagella, but it's just a budding cell that does not have a stalk, okay? So there are differences with regards to that growth cycle. And regardless of how you decide to divide, the growth rate of a microbe is very important to understand, because that gives us an important insight into how communities of microbes evolve, but also how different environmental conditions or treatments with antibiotics can affect their growth rate, right? If an antibiotic is very effective at slowing the growth rate, then we could move it forward for clinical experimentation and trials. If we see a certain nutrient enhances growth rate, that might be important because maybe we are using microbes to create a certain protein of interest for vaccine development or pharmaceuticals, synthetics, well, not synthetics, but manufacturing industries. And so when we think of the growth rate, this is basically defined as the rate of increase in cell numbers or biomass. And this is directly proportional to the population size at a given time. And so this growth rate is referred to as an is referred to as exponential because it generates an exponential curve, a curve whose slope increases continually, right? So this is integrating a little bit of mathematics into our discussion here. And when you have cell division that occurs by binary fission, so 1 becoming 2, the number of cells is then directly proportional to 2 to the nth power, where n stands for the number of generations. Now, some cyanobacteria are able to divide by multiple fission, in which case this would not apply. That is instead going to require a different set of calculations. But in general, so the default logic is 2 to the nth power will give you a clear analysis of this process. So what do you call cells that attend a religious service? Or what do you call the religious service that cells attend?
Wait, what was the question again? Could you repeat it?
What do you call the religious service that cells attend?
We don't know.
It's called biomass. Yeah, there you go. I just thought of that. I mean, like, biomass was staring me in the face. Had to do it. Moving on to talk more about biomass. Exponential growth, unfortunately, as cool as it is, it doesn't last forever, okay? And that is simply because after a certain point, at least in a closed system, you're going to run out of nutrients. You can model these effects by a changing environment where you culture bacteria, and this is referred to as batch culture. And a batch culture is able to generate a large quantity of the bacteria, but once you run out of the nutrients, then it will peak. And you can see the distinct phases here. First, you have a lag phase. This is where the bacteria have just entered into the new environment. So maybe you had a bacterial stock that was in your freezer. You take it out, not your like home freezer in a laboratory freezer. Please don't keep bacteria in your home freezer. You take it out of your lab freezer and then you put it into culture, or you get some from a plate that you had grown. So we talked about agar plates last class. You put that into the culture medium. The bacteria are not going to just start like growing exponentially right away. It's sort of like if you're not a morning person like myself, when I wake up, I don't want to do anything. I'm just like, oh, right, I'm waking up right now. That's sort of what bacteria are doing in this situation. They're like, where am I? What am I doing? Why am I here? Play Destiny's Child. Perfect. All right. So like you just sort of like getting adjusted. Then you transition to log phase where you're like going super fast, growing exponentially. At a certain point, you're going to start to run out of nutrients. That's where you hit stationary phase. So it's not that you're not moving around, but you're definitely not growing at that exponential level. So that sort of peaks. And then last but not least is this death phase where cells slowly run out of nutrients and then we'll just die. Always a little awkward, but yeah, nothing lasts forever. So there you go. And so these changing conditions impact a number of different regulatory pathways in bacteria, including their physiology, transcription factors, protein expression. And this all is integrated into the way in which bacteria can adapt to their environment. And similar to bacteria, you can think of how humans try to their best to adapt to their environment and thrive in their environments. Now, there is also a setup referred to as continuous culture, and this is where cells in a population achieve a steady state, and this allows a more detailed study of bacterial physiology. This is important because if you think about bacteria in that growth curve I just showed you, right, their expression, their life regulatory functions are going to be modulating, they're going to change because their environment is changing. Ideally, if you're able to keep a steady state environment, then you can study bacteria that are just maintaining. They're in what you might even refer to as a type of homeostasis with their environment. The environment is constant, therefore they can be constant as well. And there's a device called a chemostat that ensures logarithmic growth by constantly adding and removing equal amounts of culture media. And this is very useful to investigate from a research perspective because often the human and gastrointestinal tract is thought to function similar to a chemostat, right? We're ingesting and excreting things all the time. And so in our intestinal tract, we have this relatively constant environment where bacteria exist and will thrive, survive, or perish depending on different things, a range of different factors that we'll be discussing throughout the course. And so here is an image of both our gut and the chemostat. And these are, as you can probably imagine, fairly expensive devices. And If you're running this continuously, right, then it can get expensive quickly because you're constantly infusing nutrients, you're constantly removing nutrients, you're constantly powering this, right? It's basically trying to replicate the human gastrointestinal tract. What's interesting, though, is that you can make a number of important observations about bacteria, their accumulation or biomass, right, in these steady state conditions. And then you can also influence the rate at which they grow or get washed out based on a number of different parameters. And these relationships are illustrated in this graph here. You can see that as you increase nutrient availability, then you also increase biomass. As you increase the flow rate, so the amount in which you are washing out bacteria, right? The speed at which you are putting in, you're increasing the speed in which you're removing things. At a certain point, the bacteria will no longer be able to grow and divide fast enough, and they'll actually get washed out. And then you can also see how this correlates with generation time. And so these factors are relatively constant when you have these steady state conditions, but if you push it to one extreme or another, then you start to tip the scales. And so the graph here sort of reflects what I just said. I also say this explicitly up above here, right, with regards to these relationships. And just to reiterate, as this pattern continues, it will maintain until this dilution rate exceeds the division rate, right? So this graph will not look the same for every bacterial species or microbial species. Depending on how fast you divide, which is related to how many nutrients there are, you will either increase, maintain, or decrease your bacterial mass. And so adjusting just one of these factors can have a dramatic impact on what the steady state bacterial mass looks like. When we think about this from the specific lens of a certain microbe, then that also can change the parameters, right? So you don't see any numbers here, right? You just see that there are general trends and how different factors impact one another. If we think about E. coli under optimal growth conditions, it can divide every 20 minutes, which is crazy fast, right? Like E. coli are a great lab workhorse, so to speak. They grow quickly under these optimal conditions, so you can produce a lot of a certain protein if that is what you're interested in doing. And my lab uses E. coli to do the generation of toxin production, for example, and they're very efficient at doing that. So before we move on to this next section, are there any questions about the chemostats, the different factors at play, or the religious services that bacteria attend? Okay, that sounds good to me. So we're going to switch gears now to think a little bit more about the environments that microbes need to interface with. And these environments have a number of specific terms that dictate the pressures they face, right? So we're going to first go through some definitions and then integrate them conceptually into different environmental factors that microbes need to navigate, right? We have to navigate our own environments. The same rules apply for microbes. So hopefully some of these terms are familiar from general chemistry, but what would we define a solute as?
Being dissolved.
All right. It's something that is dissolved. Okay. And what would we dissolve it in? What would we dissolve it in?
A solvent.
A solvent. Okay. Great. Also solution, but both work. A solvent or solution. This is the definition that the textbook gives. So I'm just being sort of consistent with that, but a solvent would also work if you were to write that on the exam. What about a solution? An answer to a problem. Alternatively, the dissolver. I like it. That sounds like I don't know, a play on the Transporter or the Terminator, the dissolver. Coming to theaters when the pandemic is over. So right, it dissolves the solute. Great. So these two sort of paired together in terms of their relationship. Another important term that we'll be discussing in detail is water activity. So this would be defined as the measure of how much water is, and please do not say active, because that is, well, I mean, it sort of gets at it, but it's not what we're going for. Someone's in the chat's gonna be like, water activity is hydration or swimming or water polo. What is water activity? Available for use. Yes. That is correct. We, in this context, will use the term water activity as how much water is available for use for our microbes, right? If we are bacteria, we put on our bacteria, membranes, etc. What would our water availability look like? That's going to depend a lot on the solution that we are in. Okay, so how much water is available to us? And specifically, at least from a micro bio context, when you have water bound to ions or other solutes, that is usually not available. All right, so this relationship in terms of water activity, solutes, and solution dictate some of the pressures that we'll face as a microbe. And so it follows from this set of definitions that the more solutes in a solution, then the less water that there is available for microbes. So not great if there are tons of solutes and you really need water, okay? And so we can think about this relationship in terms of water activity by a simple equation. We want to figure out how much water activity or what the water availability is, and we need to measure something with regards to the solution versus pure water to calculate this. What is that term? What do we need to measure? What metric do we need to measure of a solution as well as pure water to figure this out?
Ions?
The ions. So it's involved with this. It's involved with the ions. The concentration of the ions. Okay. These are getting at the concept that you would measure. Solubility, important. Let's see here. I don't know if there's another way I can get it at this, but it is a type of pressure, basically, that, okay, partial pressure. All right, we're all heading in the right direction. Vapor, oh my gosh, amazing, good job. So vapor pressure blowing up in the chat, right? So basically all solutions, not just water, but all solutions have vapor pressure. And this is defined as the pressure that is generated by a vapor when it is in thermodynamic equilibrium with its liquid or solid form. All right, so this is important when we think about the microbial environments of the cell, right? And while your book does a decent job talking about this, I wish they had some more visuals that really illustrated it. So I went ahead and made some of my own to try and walk everyone through this conceptually. All right, so often we think about having a glass of water or coffee, whatever. Okay, so it's in this context, it's easier to think about water really as a bunch of water molecules, right? So, and those are made up of atoms and so on and so forth. But that's what you should think of for this context or concept. Is it's made up of a bunch of water molecules, and we want to think of this in a closed system, so we can measure the water activity. But basically, in a closed system, when you have equilibrium between the when you have water evaporating and condensing back into water, so condensation, you have this equilibrium event occurring. All right, and this is what we'd have with pure water. Okay, you just when everything is in equilibrium, then this state is fluid and it can go back and forth at an equal rate. So the evaporation and condensation is happening at an equal rate. And so when you have pure water and you're trying to figure out the water activity, what would you say the value of that is? If you're trying to figure out the pure or the water activity of pure water. Any guesses as to what is the water activity of pure water? Not Walter, water. It's a number. It's a number. Is it one? A couple. Jason and Jeanette of Destiny's Child chiming in. Yes, it's one, right? You're dividing pure water over pure water. It's going to be one every time. You can do it on the calculator. You can do it by hand. It's going to be one, right? Because you would basically just be putting the pure water on the top and it'd be divided by pure water on the bottom. So it's going to be one. And that's the maximum water activity you can have. You can't get any purer than that. All right. It's pure water. It's going to be one. So everything else is going to be less than one. And that makes sense because everything else is going to have some kind of ions or solutes in it because it's not pure water, right? So it means that everyone else or everything else will have a lower water activity for that. And in this example, you can see that there are a bunch of solutes here that are binding water, which make water molecules less available. Thus, the water activity will be something less than one, okay? For example, we could take the vapor pressure of seawater, which is 0.91, okay? Now, I'm not going to ask you to memorize a bunch of random vapor pressures, but if I gave you vapor pressure of a substance and vapor pressure of pure water, I would expect you to be able to calculate that out or determine what has the higher water activity. And basically, you'd just be using this equation. Now, water activity, as we've discussed, is a measure of how much water is available for use. And most bacteria require activity levels that are greater than 0.91. But there are still bacteria in the ocean that can deal with these lower activity levels, whereas fungi can tolerate activity levels that are as low as 0.86. Okay, so depending on your microbial way of life, you might be able to handle water that is less available, or you might have some stricter requirements, and that can have to do with a number of different mechanisms you've developed, right, to navigate that stress. But we're going to go through some more specific examples of how these changes in water power can affect microbial life. And to truly understand this, we need to introduce the concept of osmolarity, which measures the number of solute molecules in a solution. So it would follow from that if this is measuring the number of solute molecules in a solution, that it is inversely related to what concept? And I can put the question in the chat. What is osmolarity inversely related to? The answer may surprise you. Water activity? What? What are the chances that it's inversely related to what we just discussed? Yes. Good job, Domini. It is. Oh, wait. No, it's not Domini. I think it's Domini. Domini? Yes. Sorry. Please keep correcting me. I didn't come here to mispronounce a bunch of names. Let me tell you. All right. Thank you, Domini. Yes, it's inversely related to water activity. Okay. And this makes sense because if water activity is measuring the amount of water available to me, which my water activity is very low, like all I have is coffee available to me right now. So if water activity measures the amount of water that is available and osmolarity is measuring the amount of solutes, right? Those are the opposites. So it makes sense that they're inversely related. Thus, the more water particles, oh my goodness, sorry, particles there are in a solution, the blank, the osmolarity, and the blank, the water activity. What would we say here? Is it the more, oh no, wait, is that? Whoops, okay, sorry, I forgot to fix that. Don't worry about answering that. Just know that the more solute molecules there are in a solution, the greater the osmolarity and the lower the water activity, okay? So I was thinking of how to word it and I didn't edit that last slide. But basically, if you think about this relationship, the more solute molecules there are, the greater the osmolarity and the lower the water activity, right? Conversely, you could say the less solutes there are, the lower the osmolarity and the greater the water activity, right? They're inversely related. One goes up, the other goes down. Why is this relevant? Why are we talking about this, you might ask? Because like you came here to learn about bacteria and not water. Well, a cell's membrane is semi-permeable, all right? So if it's semi-permeable because of the plasma membrane, then this flux in water, the osmolarity levels, et cetera, will have a huge impact. And specifically, the membrane allows osmolarity to be different both inside versus outside the membrane, okay? So because the membrane has this selectivity, it helps maintain certain osmolarity differences between a cell's environment and its cytosol. So the cell's membrane, right, allows water to pass, but not solutes, right? So we talked about transport mechanisms in our last class, and this has a huge impact on a cell's viability due to what we call osmotic stress or pressure. And so here are some sort of basic examples thinking about membranes, okay? And based on this, excuse me, if you have a cell that is in what kind of medium, one that has greater concentration of solute particles, water will leave the cell in an attempt to equalize osmolarity across the membrane. So what kind of solution or medium would cause the water to leave the cell? Hypertonic for days, chat blowing up. Great, excellent. So we see it right here, right? Tons of solute particles outside, water rushes out, membrane shrinks. End scene. Okay, that's what happens. If we do the flip side, right, then hypotonic would be what causes an influx of water, right? It's like, whoa, super concentrated water's coming in. This increases sort of like a balloon. And if additional water keeps coming in, the cells could lyse. So these kinds of stresses are things that microbes have to navigate every day, every hour, right? They're changing environments with regards to osmolarity. And as I've mentioned, I'd like to integrate primary research into our talks, into our class, into our classroom. This exists in the real world. People study this. Here's a recent paper looking at what happens if you treat bacteria with either hypo or hyperosmotic shock. This part of the figure is looking specifically at hypoosmotic shock. And what they're showing here is if you have an influx, right, of water molecules, then at a certain point, your membrane is going to rupture, and that's the end. Once your membrane ruptures, it's really hard to make a comeback from that, okay? And so you have this rupturing, you lose intercellular components, it's really bad. But if you have these handy dandy mechanosensitive channels where you're like, hey, let's just get rid of some of these molecules to prevent the water from coming in, right? Then you can, these solutes, then you're going to be able to survive. And they looked at a specific protein channel called MSCL. And this is an example of a mechanism to deal with osmotic stress. Aquaporins are membrane channel proteins that allow water to traverse the membrane much faster than by diffusion. Okay, so this is another way in which microbes can adapt to osmotic stress. And they basically can help facilitate water traversing the membrane. And this basically is a speeding water right through the pore. So these are all strategies microbes employ to deal with an increase or decrease in water availability. There are some other approaches that microbes have at their disposal. So generally speaking, there are two mechanisms. In hypertonic media, bacteria protect their internal water by synthesizing or importing compatible solutes, such as proline, glutamic acid, or potassium ions. Conversely, in a hypotonic media environment, there are pressure-sensitive or mechanosensitive channels that can be used to leak solutes out. All right, so that's what we just talked about with MSCL. Outside their osmotic comfort range, these strategies are going to fail at controlling internal osmolarity, right? So it's not like they can only handle a very small window. Usually they have a range of osmolarity pressures they can handle. And then beyond that, unless they are able to evolve or happen to have acquired certain mutations, they're not going to be able to survive. So it's a dangerous world to be a microbe. Dangerous world. Under pressure. Throwback. Maybe that'll be a song for next DJ microboard. So I could have been a singer. I know. I hear it all the time. And it's just one of those things that you know, I probably majored in the wrong thing in college, but can't go back. No regrets. This is another example from the primary literature. We're talking about scientists studying bacterial cells when they generate this hyperosmotic shock, okay, and seeing how the bacteria survive. And again, don't get confused. This is hyperosmotic shock. This is the same thing as a hypertonic solution, right? So you see this shrinking here. So if you compare A to B, the light blue membrane region has shrunk, okay, that cytoplasm, right? And so then the cell's like, hey, we need to get some nutrients back in here, right? Because all these solutes are outside, so they pack them in, and then they are able to restore their normal way of life, right? And then they can go on to get some of these out depending on the environment that they face. You can also do the opposite, hypoosmotic shock, right? So I'd hate to be this cell. It's like hypershock, hyposhock. It's like, I am not having a good day, right? So this is the opposite, where now it's super concentrated and the scientists are like, yeah, we're gonna put you in a hypotonic solution. And so now the cell's like going to fill up and explode, or it's going to be like, get these things out of here quick, right? And then it will go on to survive. And you can actually see this by microscopy. If you look at the nucleoid region, it's easiest to see it here, right? Look at the shrinkage that is occurring in terms of that nucleoid region. It is so small, right? And that, again, is just dealing with the increased osmotic stress that these cells are under. And so I'll try to post this video and you can actually see cells exploding and stuff. It's cool stuff. Obviously, I'm a little biased, but aren't we all? All right, so as if that were not enough, microbes also have to deal with temperature, right? So it's like, do I have enough water? Do I have enough solute? Also, is the temperature changing on me, right? And so scientists have classified microorganisms based on their temperature, right? You have everything from psychrophiles that love 0 degrees Celsius to 20 degrees Celsius, all the way up to hyperthermophiles that, you know, it's like some like it hot, right? 65 degrees Celsius to 121 degrees Celsius, just like, whew, it's like climate change for real. And these organisms have a number of different strategies to navigate their environment, right? That's why we only find hyperthermophiles in extremely hot volcanic-like conditions, whereas psychrophiles we're going to find at the other end of that spectrum. And you can also look at how different microbes grow, the number of generations per hour that they are able to produce, and also depending on the nutrients that are available, right? So all these environmental factors contribute to understanding why a microbe is so good at growing at a specific condition, right? So everyone has their optimal condition. We might not know what that is from a human perspective, but we can spend time doing gradients in terms of temperature, nutrient availability, et cetera, to try and figure out what it is for microbes. We have a question in the chat. Are there microbes that can grow in both extremes? Very interesting question. I am not aware of any, but that does not mean that it is not possible. I would say that is unlikely because often the molecular strategies that a microbe uses to survive in a certain temperature range are structurally disadvantageous to the opposite one, right? So there are certain proteins that can be expressed to make your membrane more stable or more flexible, depending on what pressure you're trying to combat. And usually, if you're really trying to make sure that you're going to survive these super cold freezing conditions, that would be the opposite of what you'd want at the higher conditions, right? It is technically possible to have microbes that express both, but from an evolutionary standpoint, it's unlikely that you're going to have these psychrophiles all of a sudden being thrown in to an extreme thermophile condition, right? Or hypothermophile. They're not they're not going to be thrust in there unless scientists probably force them to. One does not get from the volcanic springs of Yellowstone or Hawaii to Antarctica in an hour. And well, actually, how would you do that? Yeah, I mean, I am not aware of any. It doesn't mean it's not possible, but definitely an interesting thing to think about. It could be. I think that would be a very cool research project, actually. So, good question. Next up, wouldn't it be organisms found in the desert like super cold or super hot? Right. So that is probably one area where you're going to find sort of extreme conditions, but you're still not going to get like Antarctica cold, right? You're not getting Antarctica cold, and you're also not getting volcano hot. That's still going to be somewhere in the middle. So yeah, I will do a little more research on that. Yeah, very interesting discussion. Bacteria in space. Yeah, I mean, here's the thing, like, do bacteria live in space? Good question. I'd be concerned about the radiation, but extreme bacteria environments. And if you, as a class, find research articles that you'd like to share with me on this topic, feel free. Like, I love to include them, right? So just let me know. So speaking of these extreme conditions, right? Here we have Antarctica, volcanic hot springs, things like that. And so we can think about how temperature impacts microbes that are in these different environments. And yeah, there was a student that asked about bacteria in space. There's a private message. I didn't just want you to think I randomly was like, and bacteria in space. Like, that was a question, but yes, lots to explore there. So some of the rapid changes in temperature actually have to deal with the heat shock response that microbes have, right? So you do a sudden change, and this is in part why microbes need to have such a fast response time. They need to be able to generate protein products such as chaperones that will help maintain protein shape, enzyme function, membrane composition. And this has been documented in all living organisms so far, which makes sense, right? Like, these organisms probably did not survive for thousands and thousands and all these different conditions, thousands and thousands of years and all these different conditions without having a wide range of response mechanisms to navigate these pressures. And this also comes into play when we think about the different elevations that microbes can handle, right? So different depths or heights in terms of elevation lead to classifications of barophiles, barosensitive, and barotolerant organisms because of the atmospheric pressure that they're used to navigating, right? And so once again, when you change these environments rapidly, there is going to be a limit to the extent at which you can survive, right? And if you look at barophiles, right, they have sort of this middle range, but they still are going to die at a certain point. But barotolerant, right, they're actually able to survive an extremely wide range of pressures, right? So there's just a lot of variability, and it really speaks to where you find a microorganism is going to be connected to the environmental factors that they need to navigate. If it's widespread, then it probably has adapted to survive in all those different contexts. If it's only found in a few spots, then it probably has environmental factors that are limiting it from colonizing a more diverse region of the Earth. Great. Let's see how we're doing on time. Yeah, we're gonna keep moving. Just in the interest of time. So as I've mentioned, there are a diverse range of stresses that microbes need to navigate. And one of the most common that we think about is oxygen, because let's face it, you take oxygen away from us, we're not going to be doing so hot, right? So it's always somewhat fascinating when we think about microbes, because there are some microbes that hate oxygen, all right? And there are some microbes that love oxygen. And then there's some microbes that are like, well, it can do with a little bit of oxygen, right? We're all about the oxygen, but microbes are not all about us, right? They have their own requirements. So let's see how we handle these definitions. How would we define a strict aerobe? What can it grow in? Right. So it needs oxygen, air, oxygen, easy way to remember it, right? Even though air is technically made up of way more than just oxygen, mostly nitrogen. Strict aerobes can only grow in oxygen. Microaerophiles, what are they growing at? What are microaerophiles? A little O2, right? You just lower levels, you sprinkle it in there. Not a ton, just little, hence the micro. Strict anaerobes die in the presence of, boom, oxygen, right? Yes, like they're strict about it. They don't mix with oxygen. It is toxic to them. And we'll discuss why oxygen is so toxic in a little bit. Next up, we have aerotolerant anaerobes. They grow in oxygen while retaining a something metabolism. A healthy metabolism? A complicated metabolism? Anaerobic? Chemical? Fermentation based. So we touched on this loosely on Tuesday, right? So when you have aerobic respiration, you can do that in the presence of oxygen. If oxygen isn't available, hey, you still have fermentation, you can get some energy out of that. And then lastly, you have facultative anaerobes that can live with or without oxygen, right? It's an option for them. So they are sort of able to possess the ability for fermentative metabolism and respiration, both anaerobic and aerobic. When we look at the growth zones of these microbes, then you are able to, actually, I want to go back. So there is an important difference here. I mixed up my terms. So aerotolerant anaerobes, I was thinking facultative at the time, but they have a fermentation-based metabolism. It's facultative anaerobes that can go back and forth. Aerotolerant are the ones that just do fermentation throughout. Okay, so aerotolerant, fermentation-based, facultative, they can do either depending on whether oxygen is available or not. Confuse those in my head. Now we're going to move on to growth zones. So often you can look at the ability of different organisms to thrive depending on the presence of oxygen. And I have to check with Dr. Skovrin if this lab module is included in the MICR 101 labs. This with the pandemic modified setup, but basically the closer you are to the surface, the closer you are to oxygen, then it moves to low oxygen, right? Oxygen's diffusing through the medium, and then finally there is no oxygen at the bottom. And we also do this a lot in the MICR 141 lab, where we look at a tube and then we see what grows based on this oxygen gradient. And depending on where you get the growth, that allows us to think or to determine what microbial species are present in these tubes. And so this has important implications for clinical microbiology and the clinical lab scientist training that our school is well known for. Now, as we discussed, certain organisms grow in oxygen, certain organisms do not. And part of this has to do with whether or not they are able to handle the reactive oxygen species that can be generated through a variety of different reactions that could then in turn damage the microbial species. So in this schematic here, there are reactive oxygen species that are highlighted in yellow throughout the schematic. And there are a number of reactions that generate reactive oxygen species. So the flavin adenine dinucleotide in the presence of oxygen and an electron can generate superoxide radicals, all right? And these are super dangerous to cells, and if you do not detoxify them, your DNA could get damaged, your membrane could get damaged, and if you do not start to initiate repairs, you could simply perish, okay? These superoxide radicals can also generate hydrogen peroxide through the action of superoxide dismutase. And this in turn can undergo the Fenton reaction, which generates hydroxyl radicals, okay? So all of these reactive oxygen species are dangerous for microbials trying to just make their way throughout the world. Now, fortunately for microbes, they have these different strategies to detoxify reactive oxygen species. So if they have superoxide dismutase, they're like, great, we stopped superoxide radicals, which are really bad, and just turn it into hydrogen peroxide, which is bad, but it's not as bad as superoxide radicals. Because hydrogen peroxide is still bad, they're like, hey, we're gonna use the Fenton, not gonna use the Fenton reaction. That just generates another hydroxyl radical. They express catalase or peroxidase to detoxify hydrogen peroxide into water and oxygen, or water and NAD+. Okay, so basically they're trying to make a very bad situation not so bad, and depending on what enzymes you have, this can be very helpful in navigating these reactive oxygen species. But as we'll discuss towards the end of the class, white blood cells often will use reactive oxygen species because not all microbes have these detoxification schemes to protect them from these dangerous compounds. We can study how anaerobes, right, are able to grow, replicate, regulate their various proteins and microbial structures through a few different techniques that remove oxygen, right? Because you might be wondering, if oxygen is so dangerous, how do we ever study the anaerobes that fail to detoxify it? And so we can use different reducing agents such as thioglycolate or oxyrase. So we use this for studying Streptococcus pneumoniae to help them grow. You can also use things called an anaerobe jar, where you remove oxygen because of the catalyzation of the oxygen through palladium. And then finally, you have an anaerobic chamber that can vacuum out oxygen and replace it with nitrogen and carbon dioxide. So there are a number of different strategies illustrated here where you can remove the oxygen from your environment. Now, importantly, a very simple way of doing this is to use a candle jar where you light a candle, close the lid, and then that will remove a lot, not all of the oxygen, but a lot of it. And you may also use this in Micro 101 lab and certainly in the Micro 141 lab if you decide to take it next semester. I know the professor, he's sort of crazy, tells a lot of bad jokes. His name is Dr. Adams. Moving on. Okay. Let's tie a lot of these concepts together with an iClicker. So again, these are questions I designed to reflect reflect exam questions. So if you find yourself struggling with these, please come see me at student success hours. If you're crushing them, that's a great sign. If you're some in the middle, totally fine. But I want to support you and make sure you're understanding these concepts. So you add red bacteria or gold bacteria to a buffer that contains hydrogen peroxide. You measure the amount of hydrogen peroxide, water, and oxygen after 1 hour that's in the buffer. What enzyme do the gold bacteria most likely express? All right, your answer choices are shown here. The data that you collected from your measurements shown here. And this pathway that we just walked through is shown here. So go ahead and take a look at the data. And based on these results and the differences you see from buffer to the bacterial conditions, try to deduce what enzyme the gold bacteria most likely express. All right, so there's a few parts. I'm going to give everyone 2 minutes to work on this, and then we will check back in. If you have any questions that you want to clarify, feel free to use the chat function or the microphone. 30 seconds left. If you've already finished, think about what it would look like, what these graphs would look like if they had a different enzyme instead. All right, time is up. Let's see how we did. All right, glad we are going to talk about this. So we have a slight majority with 25 votes for A, and then a pretty even spread for the remaining answers. Who would like to explain why they voted the way they did? And no matter what you guessed, people agreed with you.
I picked peroxidase because in the results there isn't any O2 units present for gold, but there is H2O.
All right, thank you, Harpreet, for chiming in. Do people agree with that logic? Do they see something else? It looked like what Harpreet was pointing out is that for the gold bacteria, there is no oxygen, but there is water. I see some agreement in the chat here. Does anyone want to counter or have a question about that observation? All right, so I don't see any questions. It's okay if you're not connecting all the dots right now, but I do want to walk us through this a little closer, sort of step by step. So when you approach this question, the first thing I would do is, I mean, there's multiple strategies. One strategy is to look at the answer choices, okay? However, there won't always be answer choices, right? Because I'm not going to be doing multiple choice, but at least one approach could be to think about what players are involved. So you could see here that there's peroxidase. Peroxidase is here. Catalase is here. Superoxide dismutase is here. Okay, so those are the three options, and then you have a combo option. So you could see where those are at and then think about what processes they're involved in. Or, and this is the approach that I default to, but I want to give multiple options, is to look at the data and figure out what the data is telling you. And whenever you're looking at data or graphs, always look at the axes first. So this first one, we have hydrogen peroxide. And then we have three different conditions. One is buffer, which is a control. Nothing is added to the buffer, which means that nothing is changing. All that we have in terms of information from the question is that this buffer contains hydrogen peroxide. So we're like, great, it contains hydrogen peroxide. Nothing happens to the buffer. And look, it has hydrogen peroxide. That makes sense. Both of the other conditions do not have hydrogen peroxide at the same level. That means that whatever is happening, hydrogen peroxide is being processed, right? Something is happening to it. And based on this question, it's probably being detoxified in some manner. So what other data do we have? We have water and oxygen. Where are these in our schematic? Well, if we look, boom, water is here, oxygen is here, water is here. Okay, we don't have any data on NAD8 plus, but what we do have is water data, and we see that in the buffer alone, there are low levels of this. Okay, but when there are bacteria present, all of a sudden we have increased levels of water. Okay, increased levels of water could be generated from the presence of catalase. So we see an increase in catalase, or catalase can take hydrogen peroxide and turn that into water. And peroxidase can take hydrogen peroxide and turn it into water. That is consistent with this decrease in hydrogen peroxide we see here. And finally, we have oxygen. Only one of the conditions has oxygen, and that is the red bacteria with the buffer. This is increased from the buffer alone condition, which indicates that oxygen is being produced by the red bacteria, but not the gold bacteria. That would suggest that the gold bacteria does not have catalase, because if you're producing water with catalase, you're also producing oxygen, and yet we do not observe any. Whereas if you have peroxidase, then you're producing water plus NAD+. So that is how you would arrive at peroxidase for your answer. Additional reflection questions would be, what would you do, or what would you graph if I asked you to predict the levels of NAD+ for each of these three conditions? So what would that look like based on the information you have? There are a number of other permutations you could think about, and what would happen if you had multiple enzymes present in a bacteria, et cetera. So I hope walking through that confirms your logic if you got it right, and helps explain it if you maybe missed a step. But these are the kinds of questions that I think are great for practicing, and will certainly be similar to the ones that are on the exam. All right. I hope you have a great weekend. It's been real. Listen to a whole album of Destiny's Child, and I will see you on Tuesday. Take care.